We can now start. How do I use my video? There. I'm not showing myself. Welcome once again to our regional virtual workshop on animal biotechnology in Asia and Oceania. We have our partners here with USDA FAST, Virginia Tech, and ISA Biotech Information Centers, and of course, CSIRO with Mark Tizard. Yesterday, we had a 3-hour workshop, which started with the presentations of Dr. Carl Ramage on modern biotech tools, such as genetic engineering and genome editing in improving animals, especially for disease and pest control, environmental protection and adaptation to climate change, animal and consumer welfare, food health, safety, and production, and for industrial purposes. A number of biotech animals developed through biotechnology were presented, which were detailed further by Dr. Tim Doran on poultry, livestock by Dr. Lercio Portuneto, and in aquaculture by Dr. Eric Hallerman. With the enormous scientific research in biotech animal product development, bringing the technologies to the farmers and large-scale commercialization, the benefit consumers need regulatory oversight. Policy considerations that would facilitate adoption and commercialization of genetically engineered and genome-edited animals were presented by Dr. Diane Ray Cahen, who discussed that effective regulatory approaches should be science-based, risk-proportionate and defensible, credible to the public, timely and predictable, appropriate for intended use, and transparent to all. Country regulatory policies were provided by Dr. Peter Tiggeson of OGTR Australia and Dr. Claro Mingala from the Philippines. In summary, creation of new innovative and safe agricultural products needs to be encouraged to address the growing global challenges and threats. This can be enhanced by crafting regulatory policies that are product-based, science-based, and risk-proportionate. And so thank you once again to our panelists and the active participation of the audience during the first day. Again, we invite everyone to actively participate by posting questions at the Q&A box. These will be read and answered later. We have a post-webinar survey posted at the chat box at the end of the workshop, which will be the basis of the certificates. So today, we will continue session 4 to be moderated by Dr. Mark Tizard of CSIRO, after which we will have the video presentation of Dr. Lai on opportunities in swine biotechnology. The discussion afterwards will tackle the session 4, the whole of session 4, as well as the presentation of Dr. Lai. So it's, let me call in Dr. Mark Tizard to start the floor and read and introduce all our speakers. Thank you so much.
Thank you very much indeed, Ola. Yes, and I thought it was a terrific day yesterday. Some great questions. We're continuing the theme of yesterday, and it's a great pleasure to introduce 2 speakers from the Asia region, because it's about Asia and Oceania that this workshop is focused. I also would like to quickly say a big thank you to everybody involved, that this is being done in the English language, and that's not the first language for many of our speakers or participants. So we're very grateful for that. So without further ado, I will hand over to our first speaker. Our first speaker is Mr. Asao Nakamura. He's the Deputy Director of Plant Product Safety in the Food Safety and Consumer Affairs Bureau, the Ministry of Agriculture, Forestry and Fisheries in Japan. Japan has obviously addressed this space of technology, and he's going to tell us a little bit about the policy directions and regulation that are happening in Japan. So, Dr. Nakamura, I can invite you to share your screen. Oh, in fact, actually, it's going to be shared by us. So, yeah, over to you, Mr. Nakamura. So when you're ready.
Hey, thank you. Hello, everyone. I'd like to thank you for giving me the chance to speak here today. Let me introduce myself. My name is Isao Nakamura, and I'm in charge of regulating genetically modified organisms and genome editing organisms for agricultural products in terms of preventing their impact on biodiversity. I'd like to explain Japanese policy of agricultural products obtained by genome editing technology from the viewpoint of biological diversity. I hope my presentation will help you understand the content and concept of the policies concerning agriculture, forestry, and fishery products obtained through genome editing technology in Japan. In today's presentation, I will explain three main points. First, the regulations and points to consider when evaluating environmental impacts of LMOs in Japan. Second, handling of genome editing organisms in the interest of biosafety. And finally, the procedure for agricultural, forestry, and fishery products obtained by genome editing technology. And next slide, please. Thank you. First, I will explain the regulation of LMOs for agricultural products in Japan. LMOs are regulated from 3 points of view. From the viewpoint of food safety, LMOs are regulated by Food Sanitation Law and Food Safety Basic Law. Food Sanitation Law is under the jurisdiction of the Ministry of Health, Labor and Welfare. And Food Safety Basic Law is under the jurisdiction of the Food Safety Commission. From the viewpoint of field safety, LMOs are regulated by Field Safety Law and Food Safety Basic Act. Field Safety Law is under the jurisdiction of the Ministry of Agriculture, Forestry, and Fisheries. And Food Safety Basic Law is under the jurisdiction of Ministry of the Food Safety Commission. From the perspective of impacts on biodiversity, LMOs are regulated by Cartagena Act, which is administered by the Ministry of Environment and Ministry of Agriculture, Forestry, and Fisheries. LMOs are allowed for distribution, importation, and cultivation, and so on, after completion of scientific safety assessment. Next slide, please. I'll explain from the perspective of biodiversity that I'm in charge of. I mentioned the Cartagena Act, but its formal nomenclature is Act on the Conservation and Sustainable Use of Biological Diversity through regulations on the use of living modified organisms. It is as known as Cartagena Act because formal name is too long. The purpose of this act is to ensure the precise and smooth implementation of the Cartagena Protocol of biosafety to the Convention on Biological Diversity, thereby contributing to the welfare of humankind and helping to assure healthy cultural lives for the people now and in the future. By taking measures to regulate the use of living modified organisms in order for the conservation and the sustainable use for biological diversity through international cooperation. The Cartagena Act, while Ministry of the Environment has overall jurisdiction, Ministry of Agriculture, Forestry and Fishery, Ministry of Finance, Ministry of Education, Culture, Sports, Science and Technology, Ministry of Health, Labor and Welfare, and Ministry of Economy, Trade and Industry are responsible for the ministries concerned in each field. Ministry of the Environment and Ministry of Agriculture, Forestry, and Fishery are in charge of LMOs for agriculture, forestry, and fishery products. Next page, please. I'll explain 5 points to consider in the environmental risk assessment for LMOs, easy animals. The first is competitive advantages. We confirm competition against wild animals for resources such as food, nesting site, habitat, etc. The second is predatory or parasitic. We check interference with inhabitation or growth of wild plants or animals by preying on or being parasitic on them. The third is production of harmful substances. We confirm production of harmful substances interfering with inhabitation or growth of wildlife. The fourth is cross-ability. We check hybridization with wild animals and transmitting inserted nucleic acid. The last one is other properties. We confirm such as indirectly effect on wildlife and by changing the ecosystem. We will confirm that there is no problem with all of these. Next slide, please. I'll talk about handling of genome editing organisms in the interest of biosafety. From May 2018 to January 2019, the Central Environmental Council of Japan discussed the handling of organisms obtained through genome editing technology. The Central Environmental Council have issued the following recommendation. First, if an extracellularly processed nucleic acid is not inserted into the host, the organisms are not regarded as LMOs in Cartagena Act. Next, if an extracellularly processed nucleic acid is inserted into the host and the finally obtained organisms contain the nucleic acid or replicated product thereof, the organisms are regarded as LMO in Cartagena Act. It is necessary to take appropriate measures in Cartagena Act. Finally, if an extracellularly processed nucleic acid is inserted into the host and the finally obtained organisms are confirmed as free of nucleic acid, the organisms are not regarded as LMO in Cartagena Act. In other words, the product base is used to determine whether it corresponds to LMO in Japan. Next slide, please. I'll explain summary of MOE notification. On February 8, 2019, Ministry of the Environment of Japan issued notification. Handling of organisms which are obtained through genome editing technology, but which are not defined as LMOs stipulated in Cartagena Act. In the notification, MOE notifies the basic policy of handling such organisms and requests other competent authorities to deal with those. The first is prior to the use of such organisms, those who intend to use them should provide information regarding the characteristics of the organisms and the result of review of the possible adverse effect on biological diversity to the competent government authorities. However, this should not apply to cases where the organisms are used in environment wherein containment measures stipulated by the ordinance under Article 12 of Cartagena Act or measures approved by the competent government agency are being taken. After the beginning of the organisms, if it is determined that they have the potential risk of adverse effect on biological diversity. The user should immediately take necessary measures to prevent the effect and should report it promptly to the competent government agencies. Next slide, please. As for Cartagena Act, Ministry of the Environment has overall control over all genome editing organisms. Ministry of Agriculture, Forestry and Fisheries, Ministry of Finance, Ministry of Education, Culture, Sports, Science and Technology, Ministry of Health, Labor and Welfare, and Ministry of Economy, Trade and Industry are in charge of their respective fields. Ministry of the Environment and Ministry of Agriculture, Forestry and Fisheries are in charge of agricultural products obtained by genome editing technology. Next slide, please. I'll explain procedure for the use of agricultural products of genome editing technology. In October 2019, responding to the MOE notification, MAF issued a notification regarding specific procedures for providing information to the MAF regarding organisms obtained through genome editing technology, which falls under administrative jurisdiction of the MAF. Next slide, please. I'll talk about contents of the MAF notification. First, MAF requests users to prepare a draft of information form and consult with MAF. We call it prior consultation. We assume user as developer and importer. MAF will study the contents. In an examination, opinion of persons with relevant knowledge and experience should be considered if necessary. Then, MAF requests users to finalize and submit an information form to MAF after prior consultation. And MAF announces the contents of the information form on the website. We accept information that may cause an unfair profit or disadvantage for the user when it is announced. Next slide, please. It is the format of the information form. There are 10 items. And number 1, name and summary of the organisms obtained by genome editing technology. The name and summary of the granted characteristics are described. And number 4, the fact that the organisms do not contain extracellularly processed nucleic acid or any replicated products thereof specified in Article 2, Paragraph 2, Item 1 of Cartagena Act. Information indicating that the organism is not subject to the Cartagena Act is described. This item is divided into parts to indicate that the organism is not subject to the Cartagena Act in detail. Next slide, please. Number 5, taxonomic species of the modified organisms. Information on modified organisms is described. Next slide, please. Number 7, modified gene and its function. Cleavage site on the genome. Modified gene information and theoretical changes in traits are described. Number 8, changes of traits introduced by the modification. Actual changes in traits are described. Next page, please. And number 9, whether there are any changes of traits of other than 8 above, intentional changes. Off target will be discussed later. Number 10, discussion on possible adverse effect on biological diversity through the use of the organisms. Discussion on potential impact to biodiversity is described. Next slide, please. I'll explain 4 points to confirmation in prior consultation. First, we confirm the organisms do not contain extracellularly processed nucleic acid or any replicated product thereof specified in Article 2, Paragraph 2, Item 1 of Cartagena Act. Secondly, we confirm characteristics obtained by genome editing technology and whether there are any unintended change in traits other than objective traits, including the effect of off-target. Off-target is existence of similar sequence to the target sequence. Finally, we check possibility on the effect on biodiversity. This is the same as the item confirmed in the assessment of LMOs. Next slide, please. I'll introduce about genome editing high GABA tomato. As the first case, in December 2020, Merck accepted and announced the information form of the tomato with a mutation in a GABA synthetic enzyme gene using genome editing technology. Next slide, please. For your reference, I'll share a link to the English translation of Cartagena Act. So please take a look if you have a time.
Thank you for your begin winding up now. Thank you.
Thank you for your attention.
Oh, with perfect timing. Thank you very much, Nakamura. That was great. And I really appreciated that. Was a really nice scene setting of the background, and then how you guys are actually approaching some of these gene edited products, and great to see, you know, the kind of process that we from outside of Japan will be expected to interact with when we come around to these products. Next, it's my pleasure to introduce to you Dr. Sachindan D. He's from the Animal Biotech Center, the National Dairy Research Institute in Kamal in India. He's principal research scientist in animal biotechnology, and he's had extensive research experience in animal biotechnology, has many patents on developed biotechnology tools, and is looking at improvement of livestock in cows, buffaloes, goats, and sheep. So, Dr. D, over to you. I think you may still be on mute.
Okay.
Yep, that's good. And you can go into presentation mode on your slide. That'd be great.
Yeah.
Swapped it. Yep, perfect. Lovely. Thank you.
Yeah. So I'm going to present a little bit about what is going on in Indian perspective. So to brief in a nutshell, what are the animal we are working on this genome modification? They are buffalo, goat, sheep, and poultry. And mostly scientists are working on the gene pathway. Their QTL may be affecting QTL, or example of such type of QTL is BMPR1B in case of sheep and goat to increase the fecundity. Another important is myostatin to increase the muscle mass. And we are in the cell culture and animal cloning stages and not yet produced as such with genome modified animal. So mostly we are working on the fibroblast culture so that we can able to go for biallelic change, and then by somatic cell nuclear transfer, it will be modified to make a complete animal, or we can go for zygote. We are approaching both the approaches being conducted in our laboratories. In zygote to change the biallelic mode, and then it will be complete animal will be made out of that. So some issues are there. Different laboratories, they are struggling with different types of problem with such kind of work. They are monoallelic, biallelic to find out their deletion in the genome and the screening process, and cell line, whether we are going for a cell line or complete animal, whether the cells are somatic cell or the stem cell, or it is we are going for the zygote or the single cell. So mostly we are working on the CRISPR-Cas based either in the ribonucleoprotein or in the vector based method. So these are some example how we are working at different level. So in cell culture level, we're trying to knock down this, knock out this thymidine kinase gene in the vero cell for the production of virus culture, and then BHK21, another host cell, to increase the multiplication of FMD virus vaccine. And this another way of doing it in the PPR. For all these three and the top, they are working on this virus production on the vaccine production level. And then to find out the biological pathway, how they are going to play a role in the pathway, a particular gene, how they are playing role in the pathway. So they are the one is the reproduction pathway, the EGR1 in prostaglandin 2 alpha in case of corpus luteum, how they are playing role, and bovine herpes virus and targeting a particular gene, how we are going to make a particular vaccine which would delete a gene. And some work is being initiated to produce a double muscle knockout livestock in case of cattle, goat, and sheep. Recently started, and we will be going for that. And knockout in the chicken or genome editing to augment the productivity in poultry and value-added eggs. Say, for example, low cholesterol egg. So these are the, I think yesterday their presentation, it was told that in case of CRISPR, it should be supported by proper reproductive technologies to make it successful for a young animal. So all this technology, I think the somatic cell nuclear transfer is taken lead in this particular type. And our National Dairy Research Institute, India like Karnal, Haryana, in India, they are taking lead in this particular aspect. And we are working on the water buffalo, which is present in the India, Nepal, Bhutan, Cambodia, Thailand. It is used for meat animal as well as milk purpose. So these are the different cloning calf generated in our laboratory, in our institute. So by using somatic cell nuclear transfer 2009, 10, 13, 14, and 15, and still it is, we are continuing. This is the latest one. The top figure is the donor bull, use this one, and another clone bull is being generated out of that all have been by the somatic cell nuclear transfer. So we are using this particular technology, CRISPR is being used. Same thing in case of same CRISPR, we are trying for a QTL in case of sheep and goat to increase the fecundity. Two genes are being targeted for this, BMPR1B, to increase the follicles, number of follicles, so that fecundity will be, number of offspring will be more. This is a FEC-B mutation, and it is going to increase thoroughly associated with the ovulation rate and litter size in case of sheep, as it is known long back. So these are some of the example of publication regarding this role of BMP1B in case of goat by using CRISPR. And another disease resistance, the Indian animals, they are known for the disease resistance. So we are trying to explain the pathway, how exactly they are going to play a role. So this antiviral activity, we are trying to knock a particular pathway genes, RIG1, and how important it is. Is it possible to induce antiviral immunity by using small molecule? So this pathway we are explaining by CRISPR-Cas and trying to generate a molecule so that the antiviral immunity can be stimulated in the animal. So next, this is a myostatin. I think all of you yesterday also a lot of animals people have shown. So SDN1, this is under SDN1 category. So we are trying to generate in case of buffalo, sheep, and goat. So this particular work project is started just now, and we are already, some cells are being modified by allelic form. So we are trying to generate the somatic cell nuclear transfer way to generate the double muscle buffalo as well as sheep and goat. This is the, thanks for your attention, and this is all about whatever it is happening in Indian context.
Thank you very much. That was great. It's a really good oversight of quite a wide range of research activities, all of which point very much in the direction of things that we are discussing and considering here. I'll quickly reach out to Ola. We had the opportunity to introduce Dr. Lai's presentation from yesterday, and it seems, given the nature of Dr. D's presentation, that it might be a good opportunity to wrap that talk in. Ola, if you're there, if we can.
Yes, that is actually the plan. So we would like to request Dr. Lai, and EJ is going to share the video or the recording of the PowerPoint, and then we can have the Q&A afterwards. and also to get everybody in session 4 since yesterday and today for the discussion. Okay, so let's call in Dr. Lai, please, and then EJ will share the recorded PowerPoint.
Good morning, everyone. My name is Liang Xunlai from Jilin University, China. Today, my topic is state of genetically modified pigs for agriculture in China. Pork is the main meat food in China, accounting for more than 65%. China consumes more than half pork of the world. The world consumes about 1.4 billion pigs each year, and China consumes about 0.7 billion pigs each year. According to Chinese population, each 2 Chinese will eat 1 pig each year. However, China has no native breeds of pigs with high productivity. So, China every year imports more than 20,000 elite birds from other countries, mainly from North America, including USA and Canada. Chinese government attaches great importance to generation of genetically modified pig strain with favorable trade for agriculture. Chinese government had made effort to set up National Special Fund for transgenic livestock, set up big pig facility for large-scale characterization of GM pigs. And in China, there are more than 15 research teams working on GM pigs. The genetic modification of pigs can be made to promote growth, improve meat quality, reduce environmental emissions, gain resistance to pathogen or other environmental threats. The first application of genetic modification is to promote growth. The first transgenic pig made for promoting growth is by overexpression of growth hormone. In this transgenic pig, significant improvement in both daily weight gain and feed efficiency, as well as a marked reduction in subcutaneous fat, had been observed. However, in this pig, many abnormal phenotypes also have been observed, including ulcer, stomach, arthritis, kidney disease, and infertility, etc. Then a Chinese team in 2015, they made a transgenic pig with controllable expression of growth hormone. They put a check-down system in the pig to regulate growth hormone expression. In this way, the pig can grow normally, and we also can see some improvement in growth rate and efficiency. But the improvement is not so striking as that achieved by directly overexpressing growth hormone. Another pigs made for promoting growth is editing porcine IGF-2 regulator elements to improve meat production in Chinese Burma pigs. IGF-2 is an important growth factor which affects skeletal muscle and fat deposition. In IGF-2 intra 3, there is a G. This G will be recognized by repressor ZBDE6, which negatively regulated IGF-2 expression. If we replace G with other nucleotide or remove it, then the expression of IGF-2 will be increased. In founder and F1 gene edited pigs, the body weight, muscle weight, lean weight increased, and the size of myofiber also increased. The second application of GM pig is to improve the pig meat quality. Our earlier study showed that expression of FDA2 in transgenic pigs promotes synthesis of polyunsaturated fatty acids. Mammalians lack the desaturases required for synthesis of polyunsaturated fatty acids, but the plant have this kind of enzyme. So in this study, F82 are delta 12 fatty acid desaturases from spinach was transferred to the pig, and the pig expressed the polyunsaturated fatty acids like omega-6 fatty acid. Another study showed that transfer FAT1 gene into the pig can switch omega-6 fatty acid to omega-3 fatty acid. Omega-3 fatty acid is a necessary fatty acid for mammalian development and growth. So this transgenic pig meat is called healthy meat. Recently, a China team made a transgenic pigs called expressing FAT1 and FAT2 in pigs. In these pigs, the endogenous produced omega-6 was used as a substrate to synthesize omega-3 fatty acid. So the pork from the pigs will be more nutritious. Our Chinese team also made myostatin-edited pigs to overcome laminase and sustainably improve nutritional meat production. Myostatin is a gene negatively regulated skeletal muscle cell proliferation. At the beginning experiment, they edited the exon 3 of myostatin, but this pig got some abnormal phenotype, like hind limb weakness. The pig cannot stand it, so it's not marketable. Then they edited exon 1 instead of exon 3. So these pigs grow normally. And they show similar feed conversion ratio as the wild type, and no dystocia or significant effects on maternal reproductive traits had been found. We can see the muscle of the pig increased, and the fat rate decreased in this pig. So the meat quality has been improved in this genetic editing pigs. The third application of GM pigs is to reduce environmental impact. Only 1/3 of feed nutrient and phosphorus were utilized from feedstock diets in pig production. Inefficient feed digestion can cause serious nutrient invasion to environment. Phytates negatively charged saturated thickening acid bind to positively charged molecules in diets such as minerals and protein, thereby reduce nutrient digestibility and increase discharge of unabsorbed nutrients to the environment. In addition, pigs are inherently incapable of digesting non-starchy polysaccharides that are primarily present in plant cell walls. In an early study conducted by a Canadian team, they made a pig salivary gland specific expression phytate the phosphorus passes undigested most important amino protein. Phytates allow the pigs to digest the phosphorus in phytate. So the total phosphorus content in physical matter decreased significantly. A Chinese team made a transgenic pig which express 3 microbial enzyme beta-glucanase, xylanase, and phytates in the salivary glands. These 3 transgenic pigs display increased nutrients digestibility and decreased nutrients output in physics. The growth performance also be improved in these pigs. The fourth application of GM pigs is to make stress-resistant pigs. This can be done by overexpressing endogenous resistant gene or introducing exogenous resistant genes or editing pathogens targeting receptor gene or introducing synthetic virus killing genes. A Chinese team made a adiponectin UCP1 knock-in transgenic pigs, which can resist cold temperature. Pig lack a functional UCP1 gene, resulting poor thermal regulation and susceptibility to cold. So the pigs, they produced exhibited improved thermal regulation and decreased the fat deposition and increased the adipose lipolysis. The genetic modification also can be used to produce antibacterial transgenic pigs. Nathrezim has broad spectrum antibacterial activities. Nathrezim has been transferred to the pig milk. The transgenic pig milk can inhibit the growth of E. coli in the duodenum of sucking pigs. Another antibacterial transgenic pig is porcine beta defensin 2 transgenic pigs. Porcine beta defensin 2 can against the actinobacillus pleural pneumonia. the overexpression of phospho beta deficient to enhance resistance to this bacteria. Genetic modification also can be used to produce antivirus transgenic pigs. Antivirus transgenic pigs include broad spectrum antivirus and specific antivirus. The broad spectrum antivirus transgenic pig made in China include transgenic pigs overexpressing mixed virus. This pig can resist many viruses, including influenza A and classical swine fever. The second broad spectrum antiviral transgenic pig is the one overexpressing Draco. These transgenic pigs can enhance classical swine fever virus and PR virus resistance. The third broad spectrum antivirus transgenic pig is gene editing pigs, which could enhance NLRP3 expression during the inflammatory stimulation. To make a specific viral resistant pigs, if you know the entry receptors of a virus, you can just knock out the receptor. Then the virus is not able to enter the cell. In China, a team have made a pig strain which knock out the virus entry receptor CD163 and PAPN. These pigs are resistant to PERV and TGV and PDCLV. If you don't know entry receptor of a virus, you can use the siRNA message to get the very specific virus resistant pigs. So in China, they have made the classical swine fever resistant pigs through locking in siRNA and foot and mouth disease virus and the porcine sickle virus type 2 into the pigs to produce the specific virus resistant transgenic pigs. Thank you.
Well, thank you, Dr. Lai. That was terrific. Again, a very broad range, this time with just the one species, but a broad range of very interesting applications that are coming through from gene technology, including gene editing and transgenics. So we'll now move to question and answer session, which will be, I think, somewhat mixed because we have both some regulatory considerations and also some technical input there, which was really nice. So I invite the panelists, the people who are presenting today, to come back on, turn your video on and come out of mute, and we'll field some of the questions that we've been receiving. So, and also please do keep posting up questions as we go through. There was a question kind of relates, is living modified organisms and the CBD considerations that are taken in Japan. And there was a question about the complexity of the assessment for impacts of agricultural animals, because the competitive advantage and predatory parasitic growth kind of considerations that are in there, they're very specific to something that is going to have an impact on other animals in the wild. Agriculture already has a defined place. So are there any additional considerations? So that's to Mr. Nakamura. any additional considerations with regards to that classification of a living modified organism, something that's transgenic, an animal, in livestock situations in Japan? Dr. Nakamura, are you there? You're still on mute in case you're responding to us. And we're still not hearing you, so it may be that you are on mute or have lost your connection.
He's there.
So I'll move on to another question then. This is to okay.
No, okay.
Now,
Okay, can you hear me?
Yes, we can hear you.
Yep,
I think.
there we go.
Thank you for your question. Thank you for your question. We evaluate and case by case assessment. So, but
yeah, it's really if there's anything different that you consider because of the Cartagena Protocol, Convention on Biological Diversity, agricultural animals will already sit within a contained space, I guess is the background of this. So the complexity of their interaction in a living environment is reduced just by being agricultural. Do you see there being any issue, or how that's assessed? Is it assessed differently because it's an agricultural animal?
Okay. Sorry. And is it okay to answer by email later?
Yes, sure. And we do have a number of questions here. We understand there may be complexities in those, and also you may want to actually look at the background. So I'll throw in a question. Yeah, I'll throw in a question then. I was very pleased to see that the regulatory system in Japan offers or even asks for a prior consultation, which is a great idea, and it's, I think, being encouraged globally. But do you have an idea of the timelines, how long it takes, how long you expect to do prior consultation, how long an assessment takes? Is there a time limit, or is it just open-ended?
We will evaluate, so case by case assessment. So there is no timeline.
Right. Yeah. No, okay. So it's just worked through on a case by case. Some countries have time limits from when an application goes in, which gives some certainty to producers or people trying to put things through. I'll switch then to Dr. D. There was a question about the rate of success that you have in cloning of buffaloes. How's that look?
It's written in the slide, is less than 5% if you have a skillful hand from the blastocyst to living animal. From blastocyst to living animal, it is less than 5%, depending on the skill you have and how quickly you can able to process the things in time.
Yep. Okay. So it's it's reasonable, it's acceptable, but yeah, still
Yeah.
not on the high side. Yeah. There was another question that was kind of related to that. It's, for India, what's the status of the SDN1, the kind of CRISPR knockout versus something that's transgenic, compared to, say, the declaration that Mr. Nakamura described for Japan?
We are just, it's for me.
Yes, yeah, for Dr. D in India, how is
In
SDN1
India,
approached?
in animal, we are mostly just started the work, not yet produced any animal of this kind by using this SDN1. All are in cell culture based for explaining a pathway, but not exactly the live animal we have produced till so far by using CRISPR.
Right. I have another question actually for Mr. Nakamura, which is about the success of animal biotechnology in Japan. Have you yet received any inquiries or pre-consultations? You may not be able to disclose what they are, but is that happening in Japan at the moment?
Sorry, I can't answer for this question because this is
Confidential
information.
Yeah, yeah. No, we understand that. I wonder if cheekily we can assume that means that you may have applications that have been presented. You can't tell us what, but there have been applications for animal biotechnology. Yeah, that's fair enough. We understand. We wouldn't want to ask details specifically. Actually, now there's a question for Dr. Lai. Again, really a great presentation. The question is around the concerns that might be raised about genetically modified pigs for food in China. What's the situation? How is that being dealt with and received?
Okay, so in China, the government support the genetic modification of pig and put a lot of money in the last 10 years. But the regulation so far, I think it's zero. And the scientists in China try to push the government to make the regulation to accept the pig go into the market. But in China, the public is very conservative to the GMO, and the government face the public pressure, so they can't do anything about that. This is even more serious, I think, than in Europe. So they just want, we made something there, and we just wait to see what will happen in the world. Then maybe they can make decision in future. Yeah.
That's really interesting. Thank you. That was a very interesting answer, and to characterize it as being even equivalent to, but possibly more so than Europe is interesting, because we can see some great advances you've made there technically in what's possible with the pig, and clearly the pig is a very important animal. So there was a joint question that was, does it seem likely any of the pig lines that you've described will make it into food production in the foreseeable future?
I don't see in near future it will be accepted in China.
Yeah, that's interesting, and that's something I guess our whole field considers is that there are things we can do technically, even with regulation improving the passage through, but if the public aren't ready for it, that's the problem. And so finding things that the public want us to do with this technology is key. Yeah, okay, that's really good. Just taking a look and seeing, someone actually said that I guess again with the pig work, Dr. Lai, that it seems that the gene editing research for disease resistance might eventually replace vaccines? Do you think that's a possibility?
Yes, I think so. If you like if we delete the intra receptor of virus, then maybe this pig can be replace the vaccination with this transgene.
And I know this is going to throw lots of questions your way, but I mean, like, again, it's something that relates perhaps to the public perception, the huge impacts we saw recently in China of African swine fever. Is that likely, you know, the potential for the technology to stop the need for mass slaughter, the loss of production and loss of economic values? Is that something that might drive the public to see the value in this? That you know, something like disease resistance, we have already seen in the field is an attractive topic to address for the public, making animals better and less susceptible to disease. Do you think that's a possible driver?
Yes, in China, actually now we also put up a lot of money on the African fever, swine fever in vaccine, but they also support the genetic modification of pig to resist this virus. Actually, I know several groups are doing that now. They try to find the receptor, intrareceptor of the virus. And you know, the last 2 years, the African swine fever made very big loss in Chinese porcine industry, and maybe 40% loss. That's a huge, actually. Yeah. So if we can make this pig to resist this virus, maybe we are changing the public attitude about the genetically modified animals.
Yeah, and particularly, you know, maybe the use of, you know, removal of a segment rather than something transgenic. So less, it's just less in the animal that stops the virus might be a good direction. Yeah, that's great. In relation to that, I mean, this was a sort of a spin back question to Dr. D in India, just with regards to, you know, what his feeling about the current state of regulation. It's a similar thing. I think, you know, Dr. Lai has done a great deal of work on pig. Your institute has obviously done a lot of work across other species, buffalo and goats and poultry. What do you think your perception currently of the state of regulation of animal biotechnology in India is, and the likelihood of products that you're actually developing there, making it into agricultural production and food?
Yeah, exactly. You are correct. In animal science, we are just the laws or these regulations are being framed right now. It is not in place. That's because we do not have the genetically modified organism by CRISPR method. We are having little bit of SDN1 type, cleaving a gene, say for myostatin. That also we have not yet produced. It is in the process. Hopefully, within a year or two, we are going to have modified organism. And regarding the question raised by, I think the buffalo project is going on here. We are just using SCNT process to multiply our high value germplasm. It may be bull, it may be cow.
Yeah. So you are hopeful that particularly at least SDN1 will receive
hopefully they will
favorable
allow
regulation. Yeah.
Regarding this, we are testing for the next generation regarding fertility status, and they are all genetic makeup, any off-target effect, any kind of epigenetic modification that we are currently working on, so that we can able to support with everything data that they are as such with the whatever animal we have used their cell or nucleus.
So just to wrap up, I'll wrap up then, and we'll move to our next session. We heard from Dr. Lai the public attitude in China. I'll ask both Japan and India. So Dr. D, what's your perception of the public's likely reception of these technologies, food produced using these technologies in India?
This is very difficult to answer right now. It will take some time, along with the many people they are against and they are in favor of this thing. So depending on the product we are going to generate, that will be answered along with time. It's very difficult to predict at this stage.
Yeah. And Ms. Nakamura, what are your thoughts? I mean, this is just a personal opinion. What are your thoughts on your sense of the public's attitude to this technology in Japan?
I think same as India and China. It's difficult to answer for me. Thank you.
Yeah, no, fair enough. Okay, so I think we are now ready for our next session. And for that, I hand over to my colleague, Dr. Halleman, to introduce our next speakers.
Okay, thank you, Mark. We come now to sessions 5 and 6. Now, we've heard of progress and policies on animal biotech in several Asian and Oceania countries. And we turn now to some more topical talks on the sorts of issues that those policies are intended to address. We have two really well-respected scientists to explain those to us. So first, we'll hear about food and feed safety assessment of biotech animals from Lisa Kelly. Dr. Lisa Kelly is a principal scientist in the microbiology and biotechnology section of Food Standards Australia New Zealand, or FSANZ, and has over 20 years experience in genetically modified food regulation. She oversees the assessment of applications for the approval of GM foods in Australia and New Zealand, along with FSANZ's strategic projects related to biotechnology, including work on new breeding techniques. Lisa regularly represents the Australian government on GM food matters and is a long-standing member of the Bureau of the OECD Working Group on the Safety of Novel Foods and Feeds. Lisa previously led the Australian delegation to the Codex Ad Hoc Intergovernmental Task Force on Foods Derived from Biotechnology. She's also participated in 2 FAO-WHO expert consultations on GM animals. Lisa?
Thank you, Eric, for the introduction. Let me just share my screen. Okay, can everyone see that?
Yep.
Yes, yes.
Fantastic. All right. So we are changing tack a little bit to go into some of the details of the food and feed safety assessment approach that can be applied to biotech animals. Just to briefly go over what I'm going to cover in this presentation. I'll start with a little bit of background, cover some of the key concepts and principles associated with this particular approach to assessment. Delve into some of the details of the assessment itself and the various areas that it considers, and then talk a little bit about actually applying that approach in practice, and in particular focus on some of the areas where that may be simplified or streamlined. So I think the first point really to make when we talk about food and feed safety assessment of biotech animals is that globally there is not a lot of regulatory experience in this area. And that includes in FSANZ. Most of the regulatory experience in assessing these sort of products is plant products, not animal products. And one of the main reasons for the limited regulatory experience is that there simply isn't many examples out there. There's really only 2, and we heard about those 2 yesterday, which was the salmon and the pig. There are obviously a number of edited animals that are in the process of being developed and commercialized. And the assessment approach I'm going to be talking about can also be applied to that group of biotech animals. However, important to point out that in some countries at least, genome edited animals may not be classified as GMOs and therefore may not be required to undergo any formal pre-market safety assessment. So just to start with a little bit of background on the approach itself and its origins, where it actually came from. So the approach that is typically applied to these types of products is based on concepts and principles that were developed at an international level through a consensus process. Originally, this was done through bodies like the FAO, WHO and the OECD, and these bodies produced a number of foundational documents, for example, the OECD Blue Book, which was published in 1986, as well as various expert consultation reports from FAO, WHO, the first of which I believe was published in 1991. So the concepts and principles actually go back quite a long way and were quite groundbreaking at the time. Now, these earlier documents have since been supplanted by the Codex guidelines, which were developed over a 10-year period between 1999 and 2009 through 2 ad hoc intergovernmental task forces, which were chaired by Japan. The Codex guidelines that were developed were informed by a number of specific FAO, WHO expert consultations that were convened specifically for that purpose. And those consultations focused on, there was one on plants, one on allergenicity, one on microorganisms, and 2 on animals. Now, these guidelines were primarily developed for food safety assessment, but the same approach, of course, can be applied to feed assessment, although obviously in that case, the emphasis or the focus would primarily be on animal health and safety. So there are 4 Codex documents that are relevant here. There's the overarching risk analysis, principles for risk analysis document, which applies across plants, animals, and microorganisms. And then there's the 3 guideline documents. The animal guideline was the last to be developed. Now, these, the purpose of developing these guidelines was to encourage international harmonisation in how the safety assessment to these types of products is applied across the world. Typically, what happens is that most countries, most assessment agencies have their own national guidelines, which typically are developed or based on the Codex guidelines. What we find, however, is that in developing national guidelines that are based on the Codex guidelines, there are differences that sometimes occur between countries, so that each country's approaches are not necessarily always identical, and sometimes there can be differences in how the approach itself may be applied in practice. So just to talk about the basic approach to assessment, just at a very high level, and just a few things that I want to emphasise, which are quite important or fundamental to the approach and how it's applied. Firstly, the approach itself, which is enshrined in the Codex documents, is designed to be flexible and applied in a case-by-case manner. So in other words, whilst the guidelines themselves set out core information that is typically required for assessments, it doesn't necessarily mean that all of that information is mandatory for every single assessment. So adjustments can be made depending on the type of food being assessed and the specific type of modification or trait that has been introduced. Fundamental to the approach is comparison to a conventional counterpart with a history of safe use. It's very, the comparator, it's very important that that animal does have a history of safe use, because essentially you're using that animal as the benchmark for what is considered safe and against which the modified animal will be compared. The purpose of the comparison is to identify anything that may be different between the modified animal and the comparator. And those differences then become the focus of further assessment. Having differences themselves is not necessarily any raises any safety concern, but it just tells you where to focus your assessment, and then consider whether any of those differences may potentially introduce new or altered hazards into the food products. The assessment is intended to focus not only on the intended changes to the animal, but also any unintended changes. And in the Codex documents, there's actually quite a lot of emphasis on unintended changes and various studies and approaches that may be used to determine if any of these have occurred. I think that emphasis in these documents probably dates them a bit. I think 20 years after the fact, we probably have a slightly different view about unintended effects, although certainly many members of the public still have heightened concerns about unintended changes. The final point to emphasise in the general approach is that decisions about safety or conclusions about safety should be based on the totality of the evidence. So there's no single study that that you can do that will tell you that the food is safe. So it's the evidence derived from a number of pieces of information or studies, which together allows you to reach a conclusion about the safety of the food. So this is the components or the elements of the assessment in a bit more detail. I'm not intending to go into this in a huge amount of detail. This pretty much outlines the general approach, and it's the same approach that is applied to plants as well. There are a few differences between plants and animals, which I'll highlight when I just walk through this at a reasonably high level, but really there's four main components. One is the phenotypic considerations around the host animal and the modified animal, and the health of the modified animal. Now, this particular aspect of the phenotypic considerations is actually specific to the animal guideline. It's something that you don't see in the plant guideline, for example. And the reason for that being is that we can have very healthy plants, but those plants can contain highly toxic substances to both humans and animals. Whereas in contrast, traditionally a healthy animal is generally considered safe to eat. And this is particularly the case with mammals and poultry and certain fish species. Generally, if there are substances that are produced in animals that would be harmful to humans, there is a good chance that they would also affect the health of the modified animal. So this is considered, it was an additional element that was added into the safety assessment approach that is not seen in the plant guideline. Obviously, the molecular characterization part of the assessment can be quite important, particularly in terms of how the assessment, the rest of the assessment might be framed, because obviously through that characterization process, you find out what exactly has been introduced into the genome, or changes to the genome that have been made, and in particular, whether any new substances might be expressed. So this can help you frame the rest of the assessment, as well as also inform the identification of any new or altered hazards. The assessment of new substances, this in particular with transgenic animals, for example, will primarily focus on any new proteins that may be produced, in particular an assessment of their potential toxicity or allergenicity. But obviously there may be other non-protein substances that could be introduced, for example, interfering RNA. One of the differences between, in this aspect, between the plants and animals is the idea of bioactivity in relation to expressed substances. Because of often there may be shared physiology between humans and certain animal species that are being modified. If, for example, there's a hormone that has been expressed in a modified animal, it would be important to establish that that hormone is not going to have any effects in the human following consumption of the food from that animal. The final part of the assessment focuses more on the whole food, and the main component of this is compositional analysis of the food products. However, this may, additional assessments may also be done if there has been significant changes to the nutrient composition, in which case an assessment of the nutritional impact of those changes may be warranted. In terms of the compositional analysis, this is an aspect of the assessment that can be quite challenging for animals compared to plants, and it's certainly something that is acknowledged in the Codex guideline, just for very simple practical reasons of not being able to have at hand a lot of samples, and also not necessarily being able to have closely matched comparators. And so it can be difficult to generate a meaningful amount of data in which to undertake an assessment. The way that this can be compensated for, however, is by also considering what compositional data there is in the context of the data that has been collected in relation to the health status of the animal. So in terms of applying the approach, just 3 points I just want to emphasize. It is designed for food from animals with a history of safe use, and that is really important. If an animal has been modified, that is a species that is not one that has a history of safe use as food, then this type of comparative approach may not be a valid one, and it may be necessary to actually do an assessment from first principles rather than using this type of approach. This approach has also been designed primarily for animals bearing heritable recombinant DNA constructs or transgenic animals. However, as I alluded to earlier, it could be adapted to apply to products from animals altered using other techniques such as genome editing, with certain modifications being made to take account of, for example, the absence of, if there are no new newly expressed proteins, for example. So in terms of streamlining the approach, so I've mentioned that the approach was intended and designed to be implemented and applied in a flexible way, and to really be adjusted depending on the type of modification that's been introduced and the type of food that you're assessing. Implicit in this flexibility is that the data requirements that are actually outlined in the Codex documents, for example, may need to be adjusted, and certainly some parts of that may not be relevant to the case in question that is being assessed. And so in that situation, it's perfectly acceptable to omit certain parts of the data requirements that are outlined. It may not necessarily, and of course the Codex documents are guidelines. They're not intended to be written in stone or mandatory. They are just guidelines. But of course, at the national level, it may depend on how the national guidelines have been developed and what flexibility there may be within specific regulatory frameworks and within the policy context of the particular assessment agencies as to how much flexibility there may be there. One of the issues with applying the approach in a flexible way and to introduce streamlining measures or simplifications where necessary is because there isn't a lot of hands-on experience with these types of products, particularly animal ones. Sometimes it can be difficult to know without having that experience where it's appropriate to apply streamlining. And this can be very much a matter of judgment based generally on experience. So some of the areas of the assessment that may be simplified, and once again, and this is often the case in applying the assessment to plant products, is if you've already assessed similar products before, it may be the same trait or exactly the same construct that's been used, or it may be exactly the exact same host animal that was used. Then it's possible to bridge from previous assessments to simplify the next assessment. The other issue, the other part that can be significantly streamlined is, which I mentioned before, is the assessment of new substances. If it's just a simple edit and there is no new gene that's been introduced, there are no new proteins being expressed, then that complete part of the assessment can be omitted, and that can obviously save a lot of time. And obviously, if it's a trait that has been previously assessed before and you've seen before and you know from previous assessments that that trait or that expressed protein is safe, there's no need to repeat that assessment every single time. And then also just to refer once again to compositional analyses. This can be, I sort of think of this as streamlining almost by necessity, because you may not actually have a huge amount of compositional data or a huge number of samples to on which to do a compositional analysis. In that case, decisions have to be made, well, how can I compensate for the absence of a huge amount of compositional data, and even ask questions, is that compositional data critical to being able to make a decision about safety? So those are some important things to think about. So just to sum up then, the safety assessment approach for animals is very similar to plants. However, there's very few examples of biotech animals that are around at the moment being used for food or even close to commercialization, and because of that, there's very limited regulatory experience. Some aspects of the safety assessment approach can be more challenging for animals than it is compared to plants. But the approach itself was designed to be applied in a flexible way. And I think in that context, as an assessor, it's really important to always be asking yourself that question. What am I actually trying to do with this assessment? Where are the concerns? Where do I think the concerns are? And therefore, what information is essential to know in order to reach a sound conclusion and do a robust assessment about safety, versus what information might be interesting to know or nice to know, but may not necessarily be essential for making that final judgment about safety. Okay, and that's it. Thank you very much.
All right. Thank you, Lisa, for a very clear presentation. Participants, please put any questions or comments you might have in the question and answer box. We're going to switch gears a little bit now, and we'll start talking of environmental risk assessment for biotech animals. And we'll hear from Dr. Tim Strabla from New Zealand. Dr. Strabla has more than 20 years of plant molecular biology and biotechnology research and development experience in both the public and private sectors. He's currently a principal scientist in the new organisms team at the New Zealand Environmental Protection Authority, where he evaluates and provides expert staff advice to decision makers. on all aspects of the status of organisms as new organisms in New Zealand. This involves both genetically modified and unmodified organisms, containment applications for new organisms, and release applications for new organisms. This advice includes risk analysis and risk management, as well as the fundamental biology of the organisms. He also provides technical expertise regarding hazardous substances and biotechnology to EPA staff and managers, as well as to other New Zealand government ministries and agencies. Tim.
Thank you, Eric, for that introduction. I see if I can share my screen here. Got everything else set up. Hopefully that part will work. There we go. So good morning, good afternoon, good evening, depending on where you are. I'd just like to give you a little bit about our application pathways here in New Zealand for biotech animals. I did want to just say before I get into this, I really wanted to thank particularly Diane Ray-Kayan and Peter Thigesen for giving a pretty decent introduction to a lot of the background around a regulation of animal biotechnology, so I won't belabor a lot of those points. I also want to say, Lisa's talk was also extremely informative, and I just wanted to draw, since I'm presenting in conjunction with my counterparts across the Tasman Sea, that with a food approval, there is harmonized regulation. So an approval in Australia is an approval in New Zealand, but with OGTR and the EPA, environmental risk assessments don't cross the Tasman Sea. So there are differences in regulation on the environmental side that I'll try to highlight as I go along. So hang on a second. That's better. Right. So new organisms are what we regulate at the EPA in New Zealand. And what does that actually mean? Most organisms have been around for quite some time, but what we mean by this is new organisms that are new to New Zealand. And the photos at the bottom give you some idea of why we actually have this legislation. On the left are some of our native species that in the environment that they live in, that we are trying to protect. And on the right-hand side are some of the species that were brought in with good intentions at the time, I suppose, earlier in New Zealand's history, that have led to a number of environmental problems, particularly for our native bird species. As you can see, the rat and the possum, they're munching on bird eggs and so forth. So these have caused problems. And so what our organization is about is doing, before any new organism to New Zealand can be introduced, they have to be fully risk assessed before they can be released. And so people have to make an application to us. We assess it and then decide whether or not to release it and under what conditions. And so in the middle, there are the species that we actually have outdoor approvals for. None of these are release approvals. For the cows, goats, and sheep, this is what we call an outdoor, a development and containment outdoors. So since these are large animals, they need space outdoors, but they are still contained. And so that's not under New Zealand law a release. And then the pine trees that you see, this is for Pinus radiata, Monterey pine to Americans. And that is a field test in containment. And that's both of those projects are ongoing. So what is actually a GMO under the HASNO Act? Well, a genetically modified organism is something that's had its genetic material modified by in vitro techniques or are otherwise derived or inherited. And so that's a pretty broad definition. And in many ways, it actually covers mutagenesis techniques that were in use well before any genetic modification was thought of as a thing beginning in the 70s, as Peter talked about with the Asilomar conference. And so there are a number of exemptions in our regulations under that law as to what is or is not a genetically modified organism. Now, as Peter mentioned, you know, the European Court of Justice and the New Zealand High Court both made decisions as to the GM status of gene editing. I would say that there's some difference there, is that the ECJ's interpretation is about the primary law, and it has to do with the length of time that a particular technique has been in use. And so traditional mutagenesis techniques are exempt because of the long history of safe use, whereas they judged that gene editing was perhaps, with a shorter history, perhaps the safety profiles weren't fully understood. You can make of that what you will. In New Zealand, it was a slightly different thing. Because of our regulations as to what's exempted, it was long assumed that traditional forms of mutagenesis were exempt, and certain applications of gene editing were like inducing point mutations. And so EPA in 2013, I believe it was, actually said that yes, those would be like traditional mutagenesis. So the so-called SDN1 techniques that Peter talked about yesterday would be exempt. That was challenged in the High Court. The judge didn't rule that gene editing should be considered to be genetic modification, but rather that it did not appear on the list of exemptions, and therefore had to be considered a GM regardless. This actually threw some of the other older considerations into a bit of confusion or disarray, in that point mutations weren't explicitly, by traditional mutagenesis techniques, were also not listed. So that prompted us to undertake, in conjunction with the Ministry for the Environment here, a review of those regulations, and similar to what Australia did not too long ago and got approved. But there was a cabinet decision made in 2015 to essentially maintain the status quo. So our regulations actually do say that a mutagenesis technique not in use before the 29th of July 1998 makes a GMO. And the significance of that date is simply when that part of the law went into effect. And so this is under secondary regulation, not the primary law. So there is a bit more flexibility potentially in future changes to that. So I just wanted to clarify that. I've probably gone on a bit too long about that, so we'll try to catch up. So we have a number of approval pathways that we can give, and we have given a number for animals for containment approvals, both containment or development approvals for genetically modified animals, both large and small. And we can also give an approval for a field trial in containment. We have never done that for any animal, but we have done several for plants over the years. There's only currently one that I've already told you about that's underway. And so we have done release approvals, and what you see in the pictures on the left are basically are pictorial representations of release approvals that we have done for non-GMOs. And so that's a samurai wasp at the top left there, meant to control the brown marmorated stink bug if it ever gets here. Luckily, it has not yet. And on the right is a green shield bug, an Australasian green shield bug, which the samurai wasp does not lay its eggs in. And therefore, it got approval on that basis that it didn't affect native stink bug species. But with GMOs, as with Australia, we have never approved the release of any animal as a GMO into the New Zealand environment. We can, however, do this. I will also say that no one has ever actually applied to do that in New Zealand. So we can do this without controls. That would be an unregulated release. We've only ever done that for GMOs that are medicines, and there's only one of those. We can release with controls, and that, again, has only ever been used for medicines. And then we have with or without controls for a specific pathway for qualifying organisms, which essentially are the medicines that I was just talking about. However, those pathways are available, and I will talk a little bit more about the controlled releases in the context of a full release coming up. We have one other approval that is of interest in terms of what we've been discussing here. It's for gene-edited pigs. These are not your ordinary run-of-the-mill pigs. These are actually Auckland Island pigs. Auckland Island is a very remote island in the Southern Ocean that was used as a stopover by whalers way back in the day, 150 years ago or so. And they actually left pigs on the island, and when whaling declined, the pigs remained and the whalers weren't there anymore. So these animals have been feral on that island for about 150 years. Our Department of Conservation is trying to work on restoring that island to a more pre-colonial state. And so these pigs have either been culled or some of them have been moved and brought back to New Zealand. And the significance of these animals is that because of their long isolation, they don't have a lot of diseases that other commercially farmed pigs have, particularly porcine endogenous retrovirus, but other viruses as well. And so in the context of gene editing for human organ transplant immunocompatibility, these pigs are being researched and developed for that. So that's ongoing. The outdoor containment approval, these are both done by one of our so-called Crown Research Institutes, AgResearch, which is in the Central North Island. And they are also working on cows, goats, and sheep, as I've told you. And most of the applications here as well are not for agronomic traits, but for various traits that have potential use in medicines themselves, or but there are some agronomic ones as well. So, beta-lactoglobulin knockdown for hypoallergenic milk and high casein milk as well. So it varies, but yeah, and most of that work's being done in cows and goats right at the moment. So if you want to make an application for the release of a genetically modified animal, it's likely that it will be publicly notified as required under our law. And so conditional release approvals for new organisms are required to be publicly notified, or the unconditional release as well, or to release from containment, or even a field test. And so this, with genetic modified organisms, these are still considered by some to be a bit controversial. And so I think that has sort of made a lot of potential applicants hesitant here in New Zealand. Because one other aspect of the public notification process is that we ask for submissions, and if any submitter says that they want to be heard on this, then we are required by law to hold a public hearing. And so even if an applicant, though, even if someone says they don't want to be heard, if somebody actually says that there must, that we have to hold a hearing, then all submitters can speak, should they wish. So just some aspects, and I guess probably the most important aspects of our risk assessment. I'll get into some other aspects of what we actually do look at, but these are sort of what I would call the make or break sort of criteria that we have to face and deal with in any approval pathway. And so these are the so-called minimum standards, and I won't read them back to you, but it's really all about the protection of human health, native species, and the environment. And if we can't be satisfied that those minimum standards can be met, then we are actually required to decline the application. We can't impose controls. So this is a hurdle that has to be gotten over before we can give an approval. And again, on the left, a few organisms that are non-GMOs that have gotten over this bar are shown. So this can be done, but it just simply hasn't been done with a genetically modified organism. In conjunction with the minimum standards, the additional matters to be considered, which are also very important. We're not required to decline if these can't be met, but these are very important for us to have a very close look at, because the establishment of undesirable self-sustaining populations might lead to the ability to not meet the minimum standards, but also the ability to eradicate those organisms should that actually happen. So to come back to some of the other criteria we look at, we do look at not only at the risks, but also the potential benefits, and we're required to do so. And so it's really a bit of a balancing act, provided that you can meet the minimum standards. So we look at the environmental risk assessment, public health, we've already mentioned those, economic benefit, and that's an important one as well, and a good economic benefit case goes a long way. But we also look at societal values in general, and particularly Maori culture. So our law actually requires us to consider the views, perspectives, needs, and protection of organisms and environment that are sacred to Maori. And so that's a very important part of our consideration. But we weigh up benefits versus risks in all these categories. And if the benefit outweighs the risks, then and only then are we allowed to give an approval. Right, so just a quick bit on pathways and timeframes here. There are aspects that do have statutory requirements, some aspects don't. And so I start off with what I call the pre-application phase when we receive a draft application. This is an indefinite time period. However, it will be shorter, which depends on the quality of the initial submitted application. So the ability to fully answer the sort of questions on our application form more completely will lead to a quicker evaluation and a quicker time before we can go to what I call formal receipt. And once that's actually triggered, then we have a number of steps that we undertake. So we have to do a pathway assessment. So we have non-notified pathways. Again, if it's required to be notified, we can't do that. But we can. There are a number of pathways along the notified side that we might take it down. And so we have 10 working days to deal with that. And then a public notification, if we get to that stage, and requests for submissions, that phase takes 30 working days. And as I say, that submitters may request a hearing, which would add further to the time. So we would set a date for a hearing, and that has to be done within 30 working days of the closing date of the submission to set that date. And then the public hearing, if it is required, is undertaken. And that is heard by a decision-making committee. And if that's once that's done, then the information is analyzed and drafted into a decision and discussion in conjunction with the decision-making committee by EPA advisors. And then that decision has to be notified within 30 days of the hearing. So if no hearing is required, the decision has to be considered within 30 days, and then we would publish that decision. Right, so that's a very sort of brief overview. I thought it was going to be taking more time than I did, so I might have accidentally rushed it a little bit. But that I guess leaves us a little bit more time for questions in the Q&A. So I just want to say thank you very much, and happy to answer any questions. Cheers.
Okay, thank you, Tim. In many ways, the environmental risk assessment approach in New Zealand might be a model for many countries in the region. All right, so we come now to the question and answer period, and I'll remind participants they can put their questions in the Q&A, or if you're a member of the organizers, you can put it in the chat. I'm going to start with one from Allison Van Enendam that's directed to Lisa. The idea of a closely matched contemporary comparator for compositional analysis is problematic for large animal experiments because you may not have an equivalent comparator for a clone, for example. Why not just use the public literature on variation for milk and meat composition as the guide rails, rather than having to additionally raise conventional comparator animals, which is expensive? This would also have benefit in terms of complying with the 3 R's: reduce, refine, and replace the use of experimental animals.
That's a great question. Thank you, Alison. And basically, yes, you can. You can do away with the comparator altogether. The comparator, the requirement for a comparator is reflective of, I guess, some older or early thinking in the Codex guideline, which is still there, obviously. the way the comparison is done, and typically in a compositional analysis, is you compare your new food to the conventional counterpart. If there are statistically significant differences, you then look at those values in relation to what's normally in the food supply, what the literature range is, or whatever. So even if your value is exceeding the values for the comparator, often it's within natural variation that you expect for that particular constituent. So in my mind, and this is not just in animals, you can also do this for plants. You don't necessarily need the comparator, because really what the most important comparison is how it compares to what's the natural variation already in the food supply. As I said, it's just one of those things that is enshrined in the Codex approach, and which has been translated across into various national guidelines. That is another way of streamlining the approach for sure, and particularly if that saves costs, which obviously it will in a lot of cases.
Hey, Lisa, this brings up a follow-up. Is there any move afoot to revisit Codex? It's 20 years old now.
Yeah.
And how might that be approached?
So I'm not aware of any movement internationally or appetite to reopen the Codex guideline. I do think it is showing its age. It was developed at a time very early on in the development, in the technology development, and only a limited amount of experience in assessment, and things have moved on in that time. So I think it definitely could do with a rethink. How that would or could be done, I mean, it would not be an easy process to open that up and to go back and revise the Codex guideline. And as I said, I'm not aware that there's any appetite, but it's definitely an area that I, for one, would like to see more discussion about internationally. And I really think we need to have, based on the experience that most of us now have, particularly in relation to plants, which I also think is relevant to animals, there are some aspects of that approach that we could definitely improve. For sure.
All right. Fair enough. All right, Tim, you left a loose end in your talk that I want to follow up on. You had mentioned that there's some sensitivity to Maori values in terms of new organisms. How does that apply to GMOs or to new organisms?
Thanks, Eric. It's a good question, and it's becoming increasingly important as time goes on. The HASNO Act does require that we take Maori values into account when evaluating applications, and we actually have a Maori team in the EPA that assists with that process, and they have contacts amongst Maori across the nation, and there is a group that's known, it used to be known in English as the Maori National Network. It is now called Te Herenga, and that is a group that has interest in genetic modification and new organism release technologies in general, but GMOs can quite often occupy their time. With that, we're finding that having developing partnerships with Maori when you want to do some sort of genetic modification technology is proving to be extremely useful. Consultation and engagement with Maori goes a very long way, and if you have support from local Maori, if your work is in a particular region, or if you want to go nationwide, then you potentially would consult with Tuganinga. But developing a relationship with a tribe, or a hapu as it's known, or an iwi, which is a larger sort of federation, is very helpful, very useful, and having that kind of support goes an extremely long way in getting a positive outcome for those kind of applications.
All right. Lisa, question for you. What aspects of food safety assessment of biotech animals may be more challenging compared to plants? I mean, wouldn't a lot of aspects be easier for food animals, given that the meat of animals is generally considered safe to eat if the animal's healthy, while there might be many potentially harmful or even poisonous plants?
Yeah, I mean, I think a lot of the aspects should be easier. Having said that, my background is I've spent 2 decades being involved mainly in plant assessment, so I always come with that lens. But we actually make the assessment of plants more difficult than it needs to be. But yeah, certainly with animals, we don't have that issue with various toxicants being present there. But also, I mean, I think, in relation to the assessment of animal products, and this also goes to plants, but I'll focus on animals. I mean, we have to, remember that we're dealing with animals that we have a long experience with in terms of breeding. So they've been in our food supply for a long time. So the animals that we're modifying already come with that, that history of safe use. And I think that's a really important thing to remember. But also we have to remember that quite apart from what GM regulations there may be, there are other regulations in place in countries that relate to and support a safe food supply. And those don't go away simply because an animal has been genetically modified. So they're there always in the background and help ensure that food is safe. Really, the GM assessment is really something that you overlay on top of those processes. And I guess the question we have to ask, to what extent is that always necessary to have that additional overlay? And are the current processes that we use to ensure that the products of conventional animals are safe, is that going to be sufficient in a lot of cases?
Okay. Another question for Tim. Are there any differences between the approval processes for large and small animals?
In principle, no. I think perhaps 10 years ago, there were, because, as I say, that outdoor development approval that we did was publicly notified. And we have never publicly notified any other development approval for any other animal species, small animal species. And in fact, that pig application for the immunocompatibility is one that we actually did under a non-notified rapid approval, and that passed through with relative ease. So I do think, times have changed. The Has-No Act has not changed much. Like Codex, it's more than 20 years old now. But I think public perceptions have changed to a certain extent. And it seems that there's less controversy around these sorts of applications, and the need. Some applications, obviously, we do have to publicly notify regardless, but some there are those possibilities for development work that wouldn't necessarily be required to be notified as that ag research approval was 10 years ago, 11 years ago now.
I have a question that's directed to either Peter from the Gene Technology Office or to Lisa. Do you think it's worth highlighting that in Australia, SDN1 has not been considered a GMO under the Gene Technology Act, but may require an assessment by FSANS regarding food safety?
Do you want me to answer that, Peter?
I'm going to let Lisa answer. Perhaps I'll comment on from an Australian perspective, but I might make a general answer about that, which is a riff on the theme that I had yesterday about looking at what the definitions are. So I think what's being referred to there is we have slightly different definitions in our two pieces of legislation, and I don't think that's a situation which is unique to Australia. So I think that the theoretical outcome is that you may end up in cases where in one case the food might be subject to regulation and the organism not, or vice versa. So that's a kind of theoretical one, but I'll pass over to Lisa, who might have some additional comments on that.
Yeah, so in relation to the food situation, so an SDN1 edited animal would not be a GMO in Australia. It would be a GMO in New Zealand though. The situation in relation to food is a little bit unclear at the moment, and we are in the process of undertaking work to revise the definitions that we use to determine what is a GM food and therefore subject to a pre-market safety assessment and approval by FSANZ. We are intending to shift our current process-based emphasis in our definitions over to one that is more focused on outcomes and product. And as a result of that, we won't be making these process-based distinctions between different types of genome editing, for example. What we are intending is to or we'll be proposing, and this will be going out for public comment later this month, is that products that are produced using these types of technologies that are essentially equivalent to what you could produce through breeding will be excluded from pre-market assessment and approval. Because of that, what it could mean is that there may be some edited products, including by SD1, that might actually be captured, but equally there will be others that won't. So,
okay.
Yeah,
Case by case assessment.
Yeah.
Just to add to Peter's comment there a little bit, we do have a bit of that disconnect between food and environmental risk assessments. There are lots of foods that are legal to be consumed. They're GMOs in New Zealand, but you couldn't grow them here. There have been no approvals of those organisms for growth.
Okay. Now, Tim, towards the end of your talk, you were talking about that you look also at the potential benefits of a new organism. Mm-hmm. Well, who does the cost-benefit analysis? Would it be an economist, and who would pay for that work to be done?
Yes, there. Oftentimes, if you're talking about economic benefit, then oftentimes an applicant will hire an economist to describe a potential economic benefit case. Just speaking from experience, I used to be a researcher and worked at one of the other Crown Research Institutes, SCION, and working on genetically modified pine. And that field trial at that time did have an economic analysis, and that cost was borne by SCION, and it was presented by SCION to EPA. But economic benefit is only one aspect of the case, and environmental benefit is obviously very important. And I think an environmental benefit case is probably more important in getting something over the line. Consumer benefit, which tends to economic, but if you have health benefits or something like that with a food that you might want to grow, whether that's a food animal or a food plant, that would probably carry quite a bit of weight as well. So it's quite the complex mixture. But yeah, and generally those costs are borne by the applicant, but on the other side, our fees are not all that expensive compared to some of the other costs that would be incurred with that.
Okay. In an earlier question, we were talking about some of the issues with doing comparisons of compositional analyses? This is a question for Lisa, especially because your sample sizes might be large or the animals are valuable in the case of cattle. Can animal toxicity testing be used as a substitute for compositional analysis?
There are certainly some countries where animal toxicity testing is included as part of a food safety assessment. It's not something that we do in FSANZ, have ever done, in fact, or have ever required. My view is that no, I don't think animal toxicity studies would be of value generally. You know, I think we have to a lot of times those animal toxicity studies are really done to provide reassurances around unintended effects. And indeed, that's often what the compositional analysis is used for as well. I think we actually need a more rational debate around unintended effects and the extent to which we need to be obsessing with them in terms of our assessment. You know, I've been involved in this game for a long, long time, and many assessments of plants, and I haven't seen a single unintended change that has raised any safety concerns. So if the animal toxicity testing is being done to chase down unintended effects, I don't see any value in that. And in fact, I actually don't think they would necessarily, a negative result, I actually think is going to be meaningless, because I don't think it necessarily would have the sensitivity to pick up any types of changes anyway that may be of concern. So, so in short, no.
Okay. Tim, a follow-up on the benefits question. You mentioned that it's not just money, that there could be environmental benefits and such. Is part of the consideration the distributional aspect of it? Who gets those benefits?
Right. Well, as I already alluded to, direct benefit to the environment is also of great help. You know, New Zealand is dealing with a number of diseases to its native flora particularly. I know this is an animal biotech workshop, but I will just give this as an example. One of our most iconic trees, the kauri, which I suppose would be the New Zealand equivalent of a redwood for an American perspective, you know, quite big, long-lived, majestic trees, but they are suffering from a Phytophthora infection that is sort of decimating the population. And there's a lot of discussion going on, you know, similar to what's going on with the American chestnut project about, you know, potentially doing genetic modification to make kauri resistant. I think we've got a long way to go with that, but that is one aspect. You know, I already mentioned, you know, getting support from Maori. Maori, you know, they're economically disadvantaged pretty substantially in New Zealand in a lot of cases. And, you know, and going back to my previous employment experience, working with local hapu there, local tribes, you know, the reciprocity in terms of, with regard to support was potentially providing jobs and education to Maori about some of these aspects of biotech. And that's an ongoing process, and it's evolved, I think, for Scion into a bit of a partnership. So it can take many different forms, I suppose, would be the short answer. But yeah, this is, like I say, this is an evolving story, and you know, New Zealand is not, as a, you know, we haven't released anything other than medicines, so there is still some way to go with that.
All right, thank you. Ernest Murich, you have your hand up. Please unmute and ask. Ernest still with us? Okay, well, no response, so we'll move on. So we've heard in reasonable detail about the food safety review process in Australia and the environmental safety review process in New Zealand. Yes, of course, there's issues, but I think to sort of wrap up this Q&A session, I would point out that these are reasonably well thought out regulatory systems. And I think that there are a lot of experiences and such that are useful to all of the regulators in the region and globally. So let's take our lessons from Australia and New Zealand. And I'm ready to turn over the floor to the moderator for the next session. Mark, the floor is yours. Thank you, Lisa and Tim.
Thanks very much, Eric. Yes, and thanks, Lisa and Tim. And I'll pick up just a quick theme from Tim around the traditional owners of the land from which he speaks to us. And I'll just point out that I'm speaking to you from the land of the Wathaurong people of the Kulin Nation here in Australia, in the particular region that I live in, and I pay my respects to their leaders, past, present, and emerging. It's a traditional thing that we would do in respect to the fact that we are migrants, immigrants to the lands on which we live and work. And that's the segue to our next speaker, Dr. Alison van Eenenum, who is herself a migrant from Australia, an immigrant to the lands on which she now lives in California. She undertook her biotechnology, her bachelor's degree in in agricultural sciences at the University of Melbourne. She now lives in California in UC in Davis, and she works at the University of California, Davis, the Department of Animal Sciences, and she is a cooperative extension specialist, particularly in the field of animal genomics and biotechnology. She's got a wide range of publicly funded research and outreach programs that she's been involved in over the years, particularly in animal genomics and biotechnology in livestock production systems. And recently, a lot of her work has focused on cattle. She's given more than 700 invited presentations to audiences globally. So I'm looking forward to a very smooth but impassioned presentation from Alison. She's also appeared in many forms of media, including the TV, the Dr. Oz show, Science Friday, the Intelligence Squared debate series, where I believe she won with her partner a debate about the use of biotechnology. And she's also appeared in one of my favorite documentaries, Food Evolution. If you haven't seen it, it's brilliant. It's narrated by Dr. Neil deGrasse Tyson. So a very lovely presentation and narration, but great content, particularly around this subject. And so without further ado, I'll hand over to Alison.
All right. Well, thank you very much, Mark, for that introduction. And I would like to really acknowledge Ola and the organizing committee for inviting me here to speak on this topic. Just want to double check I'm sharing my slide deck because I don't see it coming through. Is that showing up for you guys?
Not at the moment.
All right.
Yep, here we go. Now we're in presentation.
Yep.
That's it. And then swip.
All right.
And away.
Good,
beautiful.
Good for you? All right.
Yep.
Okay. So yeah, thanks for the invitation. It's been really interesting, and I'm a little trepidatious to finish out the conference because I think that this is such an important topic. And really, my training is molecular biology, not science communication, and so talk about not an expert in this area. But I did want to share a little bit of my experiences working in this field for, well, almost 25 years now, because I have watched how the public discourse around GMOs in plants has really precluded the use of this technology in animal breeding. And it's become evident to me that we have to change how we have these discussions, or we're going to continue to not be able to use this technology in livestock improvement programs, which is where my real passion lies. And so I'm going to, in the spirit of science communication, talk a little bit about a story, a personal narrative here. And this cow in the image here is part of my narrative. And for those of you that don't know, this is Princess. And she is the offspring of a genome edited bull that was edited to be hornless. And she's standing alongside her comparator, conventional comparator animal, a horned Hereford, that I just found out I didn't need to raise. Thanks, Lisa. But you can see a very obvious phenotypic difference between these two animals in that one has horns and one doesn't. And I kind of became involved in this project as a result actually of a USDA grant to Recombinetics. And they obtained funding to actually do the genome editing work and produce these two bulls that were basically genetically not growing horns and would pass that trait on to their offspring. And the reason that I was asked to participate in this project was not to do with the editing, but because my program is about extension, which is basically kind of part of the land grant mission in the United States, where there are scientists that work with universities to take science to historically farmers, so take it to the public. It's kind of Norman Borlaug's classic line, take it to the farmer. That's kind of what extension does. And so I was interested in what would industry do when faced with the possibility of, for example, having elite Holstein or dairy genetics that also came along with the characteristic of not growing horns, because typically elite dairy animals do grow horns. It's something that came along with the elite genetics. And so typically those horns are physically removed. And I want to kind of draw your attention to the timelines here because it is kind of important. So these animals were born in 2015, and this is them at Davis in November of 2015. And this publication came out in 2016. But in 2016, I actually went to an organization called the National Association of Animal Breeders in the United States, who basically have a meeting every year. And I took my clickers along and asked them a series of questions about this kind of emerging technology of genome editing. So just to get a feel for who all was there, I asked, who are you? And basically, the artificial insemination industry, not the artificial intelligence industry, the AI or artificial insemination industry, and the allied industries that go along with that. So veterinarians, perhaps, very, very small amount of producers. And this room was maybe, I don't know, 200 or 300 people or so. So this was really people that work for AI companies, or they're the inseminators that are out in the field that are doing insemination of cattle in the United States. And so just as a kind of a start off, what traits does the industry value the most out of the 6 choices that I gave them here? And this was their response. And not surprisingly, an important characteristic for them is production of milk in the case of dairy, or beef in the case of beef cattle, but not the most important. So you see that disease resistance was actually rated above that at 31%. And interestingly, the most important trait was reproduction or fertility. And in this industry audience of AI companies, animal welfare didn't actually rate very highly. And in this case, these other traits rated more highly. So disease resistance, fertility, and production traits. And then I asked them, would your company consider selling semen from a Holstein bull that had been gene edited for polled, just kind of coming out cold turkey? So this is back in 2016. So the paper had only just come out. So it was kind of a new concept. And absolutely yes, said 75% of the companies or the participants. Absolutely no, said 20% or 4%. And maybe, said 21%. So majority said yes. And at that time, no regulation decision had been made around genome editing in the United States. This was before the FDA came out with their guidance 187, and it wasn't clear what the regulatory status of these animals would be. And so how much more on average would you be willing to pay for a genome edited polled sire? And I think this number caught a few people. And I don't know if you know that AI sires are actually very expensive. That people can pay $1 million for them. So $20,000 might look like a lot, but actually that's probably not that much. And if you had an elite sire that had the best genetics and had polled, you'd probably be willing to fork out a bit of money for that. Some people not so much, but I'm guessing that people that kind of know the value of the sires were more up on the right-hand side there. And then how much more on average do you think you would sell a straw of semen from that sire if you had that added bonus in there? And here the kind of the average was about $5 to $10. And if you actually look at the cost of dehorning to producers, the labor and the loss in animal gain and the likes, it's estimated to be somewhere in the $20 to $30 range. Depends on how it's done, and it's actually quite higher in Northern Australia, where often they're dehorning beef animals at past a year of age, and there can be quite a lot of death loss and the like. So that would be kind of how they might benefit from this. And then the question, if it's regulated like conventional breeding, which basically has virtually no additional oversight, would you consider purchasing a genome edited sire? And overwhelmingly here, the opinion was yes for both dairy and beef AI sires. And there was a small number that said yes for dairy but not beef, and then neither dairy or beef. So some holdouts right out of the gate, even if it's not regulated differently. And then if you throw that additional regulatory on there, like if they're regulated like advantage salmon, would you consider purchasing a gene edited sire? You see quite a big shift from the previous slide where people at, I think it was 92% earlier, the majority of them moved over to neither beef or dairy if they're going to be regulated like a GMO. And so regulation is actually really important for industry as to how they'll do it. And I asked them, what's the greatest factor influencing your decision to purchase a genome edited sire or not? And interesting to me, economics was a big driver, not surprisingly, so 50%. But more important than regulation was consumer perception, and that was at 36%, and then a small contribution to intellectual property. So obviously consumer perception is important, and that's part of my talk here today, although the value of the trait to industry has got to be part of the discussion as well. So this particular edit that I'm going to be talking about is a project, as I mentioned, that was in collaboration with Recombinetics, who produced these two bulls that had basically an alteration where in the case of dairy animals, this polled gene has an upstream regulatory sequence where 10 base pairs has been replaced by 212 base pairs in animals that are naturally genetically dehorned, like Angus. And so there's basically, if you do the math, a 212 base pair addition upstream of the polled gene that results in this dominant polled trait. So that's why it's big P. And so this animal must be recessive, little p. And that is what you normally find in dairy animals. And in the case of these animals, this was replaced by this 212 base pair region in the homozygous condition. And so we had the animals at Davis and we bred them to horned Herefords. So again, horned is recessive, so little p, and big P. And you don't need to know too much about Mendel to figure out that probably these heterozygous big P, little p calves did not grow horns. And that's the case. And we actually produced 6 of these animals, and we followed their health over 2 years. We sequenced them, and most recently we have taken milk and meat from all of them and done compositional analysis, basically to set, I guess, some data in the public literature to help with these regulatory decisions around what is, what do we need for these types of approvals. And so here you can see 2 of our offspring that didn't grow horns, and they are flanking either side here of my contemporary comparator control animal, which was basically a horned Holstein that was mated to horned Hereford. And so we had a bunch of these very random half dairy, half beef animals at our feedlot, which didn't thrill my beef manager. And this is a male, so these two bulls here. And then this is a female, and this is Princess here, alongside poor old Cinderella, because no one was interested in her because she just has conventionally bred and she just grows horns. So that's basically this really nice visual phenotype that you can use to describe the technology and have a discussion with the public around it. And so we've already published data on the genotypic and phenotypic analysis, and we're just now writing up the compositional analysis. But what I think I want to talk about is the fact that we've been able to use these animals to do a lot of public outreach, and there's been a lot of media interest in these animals. And so Science Friday, which is kind of like the ABC of America, so it's National Public Radio video show, came out and did a feature on these animals back in, I think this was 2016. Cornell Alliance for Science partnered with us after seeing these animals discussed at the Transgenic Animal Conference, and put out a really nice little 3-minute video that just doesn't go into the technical details, but it kind of explains the rationale for why we were doing this. Why do you dehorn? Why is genetics a different approach to this? That's been really useful for public outreach. And of course, it doesn't hurt that the calves were very cute, as all calves are when they're born. And then Wired magazine actually ended up doing a feature of Princess on the front cover of her magazine, and this was in 2019. And this is actually one of the pictures they took, and it was actually pretty funny because they approached me to do this photo, and they're like, we'd like to get your cow into a green screen room. Like, it's a cow. I don't think that's happening. But they said, well, we're going to do a photo shoot. And so poor old Princess was looking a little rough. So we brought her into the shoot and got her all cleaned up for her shot. And here she is getting her 15 minutes of fame. The photographer taking her, and we put up a black thing behind her because we couldn't really get a green screen to work. And you can see we had the photographer and their assistant, and I think there was a director and a director's assistant as well. So there were 4 people out at the feedlot taking a picture of a cow, much to the amusement of the guys there. And there she is on the cover of Wired. And there was a really nice story in there. But, I kind of looked at that image. I'm like, wow, she looks a little sinister, really, because this is what she actually looked like. And it seems like often the media tend to kind of dramatize this. And I felt a bit bad for Princess because she's not a sinister looking girl. She's a nice looking girl. But then I saw the picture they had of me in there and I was like, whoa, that's the Archangel of Death. So I guess that's just kind of what media does. But we have been able to bring these animals to our public outreach event, which is called Picnic Day at UC Davis. And here you can see we had a display, and here's Princess alongside, she was with her mother at this stage, but there was a horned animal next to her. And we had some horns that the kids play with, and they basically ran around and had an opportunity to discuss this. And during this, we had a public outreach event where we brought people in and surveyed them on their attitudes towards genome editing. So here is Princess and Cinderella. And here we are having our sit-down survey. People had clickers so they could answer questions. And I did this in collaboration with my graduate students, one of whom spoke about the breeding aspects and another of whom is works with an animal welfare specialist to talk about the pain aspects of dehorning. And then at the beginning, we quizzed them, and then at the end, we quizzed them to get their attitude to the use of genome editing for animal welfare traits. So at the beginning, and I'll show you a couple of slides like this, the yellow is what we got before we gave the presentation, and the blue is after the presentation. And this was one of the run-in questions. What percentage of animal products like milk, meat, and eggs currently come from animals that have been produced using genome engineering? And this was back in 2018. And if anyone's been paying attention tonight, you'll know that the correct answer to this question is 0, because basically the salmon hadn't yet come to market in the US and the pig hadn't been approved. And so amongst this audience of people that are admittedly attending a university open house, so probably a little nerdy, only 5% of this audience got that correct at the beginning of the talk. And I guess as an educator, perhaps as concerningly, after the talk, where we very clearly said there was nothing on the market, we still only had 26 of the respondents get that correct, which was kind of a little bit distressing. So I'm not really sure how these other people didn't quite get the 0. But anyway, I guess I'm not a very good communicator, is my take home from that. But what is interesting was, how do you feel about the use of genome editing to address an animal welfare concern? And here, strongly support, moderately support. all the way through to strongly oppose, no idea, and decline to answer. The take home here was 88% of respondents at this event were strongly or moderately supportive of using gene editing to address an animal welfare concern, i.e. polled in this particular case. And basically after the presentation, it tended to move people that were somewhat supportive to strongly supportive. People that were opposed didn't really move. In fact, a couple got more opposed as we went through the talk, and that was kind of our experience. And there's actually been a couple of papers that say that public attitudes towards genetically modified polled cattle is seen positively by the public. And this paper that came out in 2019 suggests that maybe people are more likely to support GM technologies when they're perceived to benefit the animal. And interestingly, if you remember back to the NAAB survey, the animal welfare didn't rate very highly, but I think there is one in there that does rate highly in probably both audiences, and that's disease resistance. And it seems to me the messaging around the types of things we're working on has to include things that resonate with kind of the values of the general public. And I do think disease resistance, whether it's African swine fever or PRRS or especially respiratory disease resistance at the moment, might get a lot of traction. And I wanted to show you guys a little fun piece of SciComm I did recently, because we were fortunate to have 2021 be the availability for the first time ever of a genetically engineered food animal in the United States. And so just prior to the Transgenic Animal Conference in August, we had an all GMO meal. And I want to show you this video, which doesn't talk about the technology or what promoter we used or anything. It just shows good food, good times, and good products. And so I'm going to, without further ado, play you a little 1-minute video. So I encourage you to share that little 1 minute on YouTube if you want to share it with friends. But I'll just finish with this. The most desired market, aquacultural, not aquaculture, excuse me, aquarium fish in the US. I think 15% of all aquarium sales is now the glowfish. Not a food application, but a product that people want. And I must admit, in full disclosure, I have 6 of them in my tank here at home. And so they have done remarkably well. It's a very successful product that people want. And so I think the narrative that the public will not accept the products produced by genetically modified animals hasn't really ever been put to the test, because we haven't had anything yet, because until now, such food products have not been available. And if the products are something people want and are allowed to come to market, the technology can be successful. And I use the example of glowfish, but also an interesting example here in the US is the Impossible Burger. It's a plant-based, soy-based burger that has genetically engineered heme to give it the bleed, if you will, the red bleed of a hamburger. And that has been a very successful product. There hasn't been pushback on that at all. And so I think if products people want are available, that this whole kind of narrative that the public won't accept it is not borne out by actual market data. And as we've learned over the past year, very painfully, science doesn't change minds, and it doesn't necessarily result in evidence-based policy decisions. And I'll just leave it at that. I'm not going to get political. But I think many potential applications of biotechnology do address traits that are of concern to the general public, like disease resistance, animal welfare and resilience, and product quality traits. And I think AquaBounty is doing a beautiful job right now of emphasizing shared values, of being able to grow that fish in the United States with a reduced environmental footprint in a sustainable way. They don't emphasize the method that was used to produce it. They emphasize the benefits and where it aligns with people's values. And I think that that's really how the public communication has to go. And it doesn't really matter how these animals were produced, but this messaging and storytelling, if you will, is such an important consideration in the commercialization of these products. And I think that's the message that we have to take home. And whose job is it to do it? I'm not sure, but I know if we don't do it, that we will not be allowed to use these technologies. And so I encourage everyone that's involved in this to do your bit in terms of trying to get the message out and to actually discuss the use of this technology and the benefits that it potentially can provide. So with that, I will stop sharing my screen and open up or pass it back to Mark.
Oh, well, in fact, open it up to questions, exactly as you're going to say. Alison, that was great and spot on to time. I loved the sharing of that short version of the video. All GM, you know, like the foods are out there now. It's starting to happen. So yeah, look, there've been a number of questions, and I invite people to continue to post them up. One of the first questions was in relation to that change in perception of the breeders. There was a great study, taking a great opportunity with interacting to get some sense from a key component in that through chain, from where here's the animals, the science, through to getting to the public. The change in their attitude when they saw there was regulation, do you think that was driven by their perception of the cost of the regulation, or their perception of the impact that would have on the perception of the product by the public? It's kind of like trying to unravel.
I think that's a really hard question to answer, and it, probably a little bit of both. You know, I think in my experience, particularly the dairy industry got hit hard by recombinant RBST, and they are gun shy of doing anything that can allow the marketers to suggest that their product is not good in some way, or, you know, basically the monetization of the fear around GMOs has created its own little, you know, market, and they don't want to have something that the competitors can use against them. And so I think there's a real hesitancy there, unfortunately, that is a painful reminder of that. And then also I do think, I mean, at that time the fish had not been approved, or maybe had just been approved, but it was still another 6 years before we had that meal. And so I think there is a hesitancy for what the cost of that might look like. And quite frankly, if it takes more than 2 weeks to approve the next generation of genome edited bulls, then the next generation will already be better, because they're getting better every year. So you can't hold up an animal for 15 years seeing if the meat and milk from a polled animal is equivalent to a non-polled animal, because your genetics of your competitors will already have surpassed you, basically. So yeah, that's a really important notion in animal breeding, is we're getting better every generation, and so you can't hold up progress.
Yeah, that's an interesting point, Alison. I was going to leap to another question, but it's worth following that through, because, you know, I think that is a significant impact in terms of how we can make benefit from animal biotechnology. And you've kind of pointed it out there, that it's this issue that fits in with polls. You can poll by just breeding with a cow that's polled, but you lose half of your values, and no one's prepared to do that, which is why we need this technology. It does it in a single generation, but those generations are constantly being improved. And so we have to figure out how we time the approvals, and it does vary from industry to industry. I think the cattle industry is one in particular where this needs to be resolved. Some other industries, that there's some steps in generation from they're improved to when they get to market, but it's still going to be difficult.
Just look at that little example I gave of the polled animals, right? So they were born, I think they were actually edited in 2014.
14, yeah.
Born in 2015. It's now 2021. We've just now finished the meat analysis of the F1. And we are doing, and that's with a USDA grant for biotech risk assessment, which was, provided the funding to do that. That's 6 years, 7 years. That's not going to work. That's the end of it. Even if it passes and gets in and everything. So I think I think it's great to do that one time, just to kind of provide some data and to have a discussion. But that can't be the standard, or else it basically will cost it out of the market.
Yeah, that does point towards perhaps the need for some different systems of how these things are assessed, so that the safety of the process, and then just some simple tick marks that you have to do on the outcome to enable that to happen in real time for animal breeding processes. Another question that came through was to do with the thermal tolerance as a trait that can be modified, and how that might be received. I mean, like that. Potentially it's a welfare of the animal to make it perform better in a different environment. But then again, the flip side of that is, well, is that just a production trait because it's, you're making the animal production animal in an environment that it's not really suited for. You're trying to tailor it to that. Yeah, is that a good trait?
You say production trait like that's a bad thing. And I think that we need to rephrase how we discuss that, because aqua bounty is a production trait, but it's a production trait that enables them to be grown locally and to do so with less feed. And, so yes, production traits have a negative connotation, but actually they're part of reducing the environmental footprint. But with regards to thermotolerance, so you're obviously well aware of the SLIC mutation that originated, I think, in Senapol cattle. That is one gene. I think, in my opinion, a lot of traits that have to do with thermotolerance that are maybe in well-adapted tropical breeds that aren't in, for the animals in our back or in my background, is going to be multifaceted. And so there are some simple single gene traits that could be introduced, but to live in the tropics, you need tick resistance, and you need a whole bunch of other stuff that maybe some of these Bos taurus animals don't quite have. And so I think it's going to be a little bit more tricky than that. But having said that, there's no reason why not to do SLIC, right? And so I think I get nervous that it gets posed as a silver bullet, but at the same time, I often use the analogy of an ice cream sundae with a cherry on top, and the editing is the cherry on top. And you can have an ice cream sundae without the cherry on top, with traditional breeding, but it's nice if it does have a cherry on top. So, why ban the cherry? So that's kind of my question.
Yeah. So, I mean, I guess that sort of speaks to this thing about what are the examples that we can take forward that will have that buy-in from the public. Because again, sort of coming back to some of the work that you did there again with the open day and seeing what the public's response is. Do you feel that the field needs those kinds of things, the best examples that we can as the thin end of the wedge to push in and open up the conversation with the public?
Certainly the visual is helpful, because otherwise it's like, here's my animal that looks exactly like this other animal, and it's hard to appreciate maybe it's thermotolerant or it's disease resistance, although I guess you could have a sick pig. But I think that was part of the attraction. I will say we did have a couple, as you probably saw, of protesters that came to that meeting, and so you do have to have your plan as to how you're going to deal with that. And what I always remember is there's a bell curve of people. There's people that are very pro and people that are very anti, and they're very noisy. And most people are in the middle of that bell curve, and they're watching and seeing how the interaction goes. And, are you being civil, and are they being civil? And are you having a discussion or a yelling match? And I think there's an opportunity there to demonstrate goodwill and make sure that you keep it at a level that the silent majority are watching what you're doing, and you have to think through how to handle the, sometimes difficult conversations that are associated with this technology.
So a question that's arisen, and this kind of relates to that, is when we talk about the public, but then for many countries in Asia, the conversations are early, and it's actually at the moment for scientists reaching out to policymakers and regulators. And I guess that I'm thinking that's where this question aims at. It's basically, how can we improve effective science communication and raising awareness on animal biotechnology to, maybe not just to the public audience, but to the audience, to some extent to us that counts, which is the people who will form that bit in between us and the public.
I mean, I think you do have to go to your equivalent of Washington, DC, whatever that might be, and be involved in these committee meetings and the types of things that was mentioned earlier, that, for example, Lisa said she was involved in this, discussion and that discussion, and actually try and have a seat at the table, and not be afraid to say when things are being, especially regulations are being proposed that are, don't make any scientific sense, because you can kill a technology with over-regulation, and I would argue genetic engineering in animals has been, and I'm very nervous to lose genome editing to the same problem, and so I think it's up to us to say, look, this is a really valuable technology, and be part of the discussion, and you have to have a little bit of security of employment. I'm very fortunate at a university. I could say what I want and no one can fire me. It's easy for me to say that, regulators should speak up or whoever, but it's like, well, if that's their job, that's maybe a little bit of a different scenario. So I do think having an independent academic that doesn't have, their job doesn't depend on it and that does have work in this area and expertise is really a helpful thing. And so I would really encourage academic researchers to do it. But of course, it's regulatory discussions are quite frustrating at times, and so it's not everybody's cup of tea. So, but I think it's important, because otherwise, why do the research if you can never use the products? I guess to me, it's almost pragmatic. It's like, I don't want to not use this technology too.
Yeah, and that's another great point. I mean, for those of us that come from that research end of things, it is frustrating now to see the applications and the benefits that could flow from this, and that we can all have a sense is going to be much needed. This being an Asia Oceania meeting, that the population growth for the planet is driven through the Asia region, and the pressure on generating high quality foods from animals is largely going to be here. There's some other big places around the world, Africa and South America, where that's happening too. But here's a space where the pressure on the system, we can see where some of the things that are available to us in biotechnology can add value, improve things, improve sustainability, which is going to be most important. Break down disease, which is important in both directions. Animal welfare, which people are concerned more about, plus the impacts on production. So yeah, it can be frustrating, but then there's also the flip side, which is regulators are put in place based around this issue of maintaining safety. And I guess it's that thing about, as scientists, we look at the science that's in this, in terms of what is not safe, versus the values systems that are driving the public perception, which impacts on the politicians who make the policies that the regulators then have to put in place. So it's an interesting spectrum that we sit in. Let's see, are there any more questions that we have up here? At the moment, no, there are no more questions. So there
is one, I think, that just came in on the questions and answer, and I don't know. Yeah. We may want to invite back some of the other speakers regarding off targets. I think Lisa made a fairly strong statement in that regard, and I do believe it's become the boogeyman of editing. So it's kind of the unknowns, and it's, I guess you have to put again in context of naturally occurring genetic variation, like that is the basis of all evolution, right? So every meal you've ever eaten has off targets relative to every other meal, and so it's become as if off targets are in and of themselves a safety hazard, and I think that it's very different when you're editing a human patient somatically for cancer treatment or something, where you might switch on a oncogene or something, as distinct from editing animals where if you do something that really messes them up, they'll never be born. And so if they're running around healthy animals, you've already done an incredibly complex experiment to see, are you a functional living being that's healthy? And that's a pretty big experiment, and lots of animals don't pass that experiment, right? And so I think that there has to be kind of an understanding that variation in your food, DNA variation in and of itself, is not a hazard, and that's distinct from potential somatic editing for therapeutic purposes in human medicine. And that's where the off-target discussion starts, and then it kind of gets carried over to agriculture like it's relevant. And I have a hard time coming up with the hazard.
Yeah, I think that's again that one of the that's a good answer to that question too, because I think there are regulatory issues that sit in the background of that, but I think they're being looked at reasonably sensibly. But yeah, at the end of the day, it's a misapprehension by everyone about what animal breeding is about, and where the animals that we eat today have come from compared to the wild creatures that were domesticated 10,000 years ago. Sex, I love giving lectures to students. I put up sex in big red letters and let the X dissolve into a chromosome shape. That's what it's all about, natural variation. And the problem that we have about identifying off targets is very often distinguishing it from background variation, because there will be variation from generation to generation. It's like, did it come from the technology, or was it natural? Does it matter? Because of the filter, as you said, the filter to get to a living animal that is born, that stands up and walks around or swims around if it's the fish, is the major filter. So yeah. There was another question, actually. Here we go. In the role of august bodies like the National Academy of Sciences or our Australian Academy of Learned Organisations, what do you think their role is in this sort of debate?
Well, I mean, the role of the national academies, and maybe similar in Australia, is really to provide unbiased scientific opinion to Congress, is my understanding anyway, but Americans can probably correct me. They're over on the East Coast, so they're probably asleep because it's 2 a.m. in the morning. But I think that they're always going to give a scientific answer, because they're going to have a bunch of science nerds on those things giving scientific answers. And to your point, that doesn't necessarily translate into policy, and that politicians are obeying a different master, and so policy may not represent that. I think you have to start with these are the facts, as we've seen. If that's not your foundation, you get some very strange decisions and recommendations that get made. At least there has been, as it relates to COVID over here. So I mean, I think if nothing else, this pandemic has kind of crystallized how misinformation can harm public health. And I guess I feel like agricultural scientists kind of like hold my beer, because I feel like we've been dealing with this for a long time, and it's just now people are going, wow, this actual marketing of fear and kind of alternative products that have dubious efficacy is actually a real problem, and it's really hard to counter with science and facts and expertise. And I think that the medical profession is facing that at the moment, and I don't think we have a good handle on it, to be honest, because we have real hesitancy around taking a safe, effective vaccine.
Yeah, absolutely. It's a warning to us, I guess, in this field, not to rely just purely on the nature of the science. And I've got scribbled down on a piece of paper in front of me here, science versus values. We always want to talk about this, but we've got to accept that there are values, and we have to face how we deal with that, and how they impact on acceptance of the things that we're proposing and that we would like to see come through. Alison, we'll have to draw it to a close because we're getting towards the end of the workshop. I'd say a huge thank you because it is actually, for those who don't know, nearing midnight in California. So you've stayed alert and awake for us, which is absolutely fantastic. Ola, I believe that it's down to me now to do a little bit of a drawing up and a wrap up, a key takeaway message from today and yesterday. In which case I'll launch straight into it. I think, my take on this, we've had, and a huge thanks on our behalf to the range of speakers that we've had. They've covered a broad range of issues and topics that relate to the application of gene technology, animal biotechnology in agriculture. And we've seen that there've been some huge steps forward, through the new opportunities that technologies like CRISPR present to us, and that we've heard a range of possibilities of things coming through. And reminder, there's only one product on the market at the moment that uses biotechnology in the production of an animal food for consumption by humans. But there's a lot, obviously, in the sidelines ready to come forward. We've heard that a great presentation there about the importance of communication, because I think communicating why we want to use this, what the value is for the public, who are the ultimate arbiters, and to everyone in the chain on the way through. The regulators are absolutely critical, obviously, for us, because we have to pass through that filter with the things that we bring on as scientists and aim towards the public. But they're there to maintain safety, and we have to embrace that and work with them. I think, it's also been clear that the regulatory systems are not standing still. It's really reassuring to see that the regulators are working almost as hard as we are to take a look at what all of these technologies mean in terms of changing the kinds of policy and regulation that was set in place 2 decades ago to deal with biotechnology coming through in plant food production. So flexibility seems to be a common theme. There's attempts in a lot of these systems to build in some flexibility. Of course, as scientists, we see this drive to safety in a different way because we look at the scientific detail of safety, but there are laws that are in place that set the scene for the regulations, and there are boundaries around that. So we have to work with that. There are reviews of things changing. Across Asia, I think the great opportunity is many countries who are looking at this and saying, gosh, we need to put some regulations in place, and that is being informed through this debate. And so for the participants that we have online, I invite you, reach out to us. I think you can see that there's a very positive and friendly and welcoming community here amongst both the scientists and the regulators in this space. There's a community that's been pulled together. ISA is a fantastic organization for doing that, for bringing these things together, and my hat's off to Ola and her team. I think a huge round of applause for them. They have done a terrific job in pulling together the program for this, and also handling all of the technical background issues to make it possible for us to see this presentation, to have people from so many places around the world all in the one room here together. So a huge thanks to Ola, and yes, just a call out to everyone. Communication is absolutely essential, not just with the public, the regulators, but between us as a community to try and take this forward. And we're looking for, harmonization and ways to bring this technology, not just in individual countries, but between countries, sharing this and sharing the benefits for us all, for the agricultural production systems, and ultimately for people, for making good, safe foods more effectively, more sustainably, just better. With that, I'll hand over back to Ola to finally wrap up. And thanks very much, everyone.
Thank you so much. It was indeed a very exciting workshop, the first in Asia, Oceania. And we have gathered a lot of panelists, experts from different parts of the world. The exchanges are excellent. And we have seen how each country have developed their own R&Ds in different animal species, and we hope that other countries can also join us in the future. We're expecting countries like Vietnam, Malaysia, and others, and we want to hear from them. Actually, what I would like to emphasize is that we are looking at some activities, some important activities in the future, because this virtual workshop actually has gained us a lot of momentum in capacity building and in science communication, in regulation, and of course, the science. So I would like to invite all the attendees to please respond to the post-webinar survey. We have questions there that we need your information or your inputs so that we can move forward. We can extend the learnings that we have learned in these 2 days. So for example, we want to have your information on what other capacity building activities that you would like, so that you can appreciate and you can benefit from animal biotechnology, and what are the who are the experts that needs further engagement or focused discussion with our experts that we have now, and also what are the gaps that your country needs. So I hope that when we receive your post-webinar survey, we would be able to discern some of these activities that will target to your needs, and we can help you out with that. So in closing, I would like to thank again all the panelists. Thank you so much to our expert moderators, Mark Tizard and Eric Hallerman, and also to all our supporters, of course, USDA, CSIRO, Virginia Tech, and ISA Biotechnology Information Centers. And well, this is really wishful thinking that we will see each other face to face. We have been meeting every other week, the Animal Biotechnology Organizing Workshop Committee, and we're looking forward for a face-to-face, hopefully very soon. So with that, thank you so much once again, and see you next time for another engaging animal biotechnology workshop in the region or internationally. Thank you so much. Bye-bye.